So hello and welcome. I am Dr. Lauren Rast. I'm the Vice President of the UAB chapter of Sigma Xi. So welcome to our monthly March meeting for the UAB chapter of Sigma Xi. That's part of our monthly seminar series. Just a few items of business. So thank you for your membership. Make sure to renew your membership. You can do so using the link provided there on the slide. And if you run into any issues, you're welcome to send us an email at the email address there on the slide. So just a couple of reminders. The 2021 annual meeting and student research conference, November 4th to 7th, 2021, has a submission deadline of Friday, April 2nd, 2021. You can use the link there to learn more. And we also have grants in aid through the national office. So the application deadline is October 1st, and that is for undergraduate and graduate student travel and research funding. And our next seminar will be on Wednesday, April 21st, 2021 at noon. So this is just a little bit more information about the annual meeting and student research conference. So the title of the conference is Roots to Fruits, Responsible Research for Flourishing Humanity, How Scientific Virtues Serve Society. And so that is at the Conference and Event Center in Niagara Falls, New York. So make sure to stay connected to Sigma Xi. You can look at for the announcements. Let us know if you want to be added to our email list. You can also follow us on Facebook, and you can send us an email as well. So that email is sigmaxiuab@gmail.com, and we also have a YouTube channel there, Sigma Xi UAB. Okay, so in February, our meeting was for Darwin Day, behind the science at UAB, integrating research and medicine to respond to the pandemic. And our upcoming seminar in April is going to be presented by Dr. Zenithia Barnett, who is a research fisheries biologist at the Forest Service Center for Bottomland Hardwoods Research at the Southern Research Station. And that is, again, on Wednesday, April 21st, 2021 at noon Central Time. And her talk will be on crayfish and dams, the good, the bad, and the ugly. So please join us for that. She'll also be talking about career path related issues as well that might be of interest to students. So, and for today, we have our own Sigma Xi president, Dr. Anna Oliveira. And so Dr. Oliveira is an assistant professor from the UAB School of Health Professions, and she's, again, president of our UAB chapter of Sigma Xi, and she got her degree, her doctorate from UAB in 2010. And so she's going to be talking to us about potential for zoonotic parasitic transmission in dog parks in Birmingham, Alabama metropolitan area. So I'm very interested in that topic from a scientific point of view and also as a new puppy owner. So I'm very, very interested to hear what you have to say today. So thanks for everyone who's in attendance, and thanks for your membership. We will have time for a few questions at the end, so make sure to put them in the Q&A box, and we'll moderate the questions for you. And again, if you want to receive emails from us, send us a message at sigmaxiuab@gmail.com. So without further ado, I will stop sharing, and I'm going to hand it over to Dr. Oliveira for her talk.
Thank you. Thank you. Excited to be here.
Welcome.
Let me see. Oops. Are you guys seeing the correct screen?
It looks good to me.
Okay. Sometimes, you know, with two screens, you kind of...
You are on presenter view, Anna.
Okay. I'll just swap. Good?
Perfect.
Okay. All right. So, thank you so much for this opportunity to share with you guys. And this, it's not a a biggest science. Those are what I'm going to planning to share with you is some work that I've done with my graduate students, my master's students here in the School of Health Profession in the clinical laboratory science program. But it's something very dear to me. I love parasitology, I really do. I teach parasitology, and I love science. So this is a way to do a little bit of research with a small budget, but it's still, learn interesting things. And, because I'm just think that also we are all scientists, we are all people, but we are inserted in environments, in a society, and we have, civic responsibilities and things like that. I just think that is so interesting how science and sociology and history and all of that intersect. So it was very interesting to me in learning about parasitic diseases, specifically in the US, how it actually mimics a little bit what I have been seeing, in Brazil. What is two different countries, right? But we can see that difference. So, so the topic is the potential for zoonotic parasitic transmission in dog parks in the Birmingham area. So I'm going to talk to you a little bit about the background. This pilot study, we actually did a part 1 and a part 2, and you're going to see why. And also at the end, we're going to try to tie it all up with, parasitic infections in the US and in Alabama specifically in the past or in the present. I told you I teach parasitology, and sometimes I have to convince my students that parasitology is important and that parasites are here in the US. Sometimes they don't think that they are. So I have to justify to make sure that they know as a clinician in the US is important for you to know about these parasites and properly diagnose them. Because at first for me, it's just because they're cool, right? But, I have to justify. So in terms of background, so we know that the number of dogs in the households has increased in the US. So the estimation is that 45 million households have dogs, right? And we also have an increase. We have seen that now more and more you have dog parks, right, in neighborhoods where dogs and people go. So here we had something on 2019 from AL.com where, was actually suggesting 32 dog parks in Alabama. And some of them are amazing. They have different things for the furry friends, right? And we know that even at the White House, right? Not only our houses, but at the White House, we have some dogs too in this. I thought it was a cute one. There is a presidential pet museum, and this is JFK's Cocker Spaniel Shannon. So we think about dogs, we think about, people, and I'm thinking zoonoses, right? Because zoonoses are those diseases that actually pass to us, that are transmitted from animals to humans, right? So there are some that are very important, like Giardia. Hookworms is one that is very important too. Toxocara, that's an ascaris of dogs and cats, canis and catty. And there are a lot of tapeworms too that can cause disease that are mainly from animals, but they can cause diseases in people too. So we have these general statistics that 1 in 18 dogs are testing positive for hookworm. We know that in humans, we had a lot of prevalence of hookworms in the south of the US, and we'll talk about this a little bit towards the end. And there are some recent studies that also kind of point to we have in Alabama, for example, also infections with hookworm. So, if you have a pet, and Dr. Rest just said that she just has a puppy. So your puppy probably have that little belly. I call the belly of worms, right? That's why it's full of worms. So they should, right, visit the vets, right, the doctors, regularly to have, anti-helmet treatment. They have the treatment for the Dirofilaria, that is the heartworm, right? And when they visit the parks, there should be some measures there to minimize transmission, right? But if this these preventive measures, right, of deworming your dog and also following the measures to avoid transmission at the park, then it's feasible for us to think that zoonotic transmission might occur. And here are the top dog names of 2020, if you need some suggestions. So the dog park is a place for all dogs, right? If you go to some parks, they are so big that they have a small dog area, large dog area. They even have sometimes dogs with disability areas. But they are also a place for people too, right? And a lot of people. So there are some measures there that already decrease transmission, right? You have the rules like no food there, keep your dog on the leash, and all of those things. Some of the things that I wanted to point out, at least this is Lockridge Dog Park in Hoover, but even in smaller ones will have some of those things. So, you have sometimes, the trash bags there, you have a trash can where you can dispose of the stools of the animals, there's water already there, a water fountain. So, and you're not supposed to bring food or anything like that. So all of this makes us think, right, just looking at that the transmission risk is low, right? But then when the rules are not followed, and they are not followed all the time, then something happens. And this was just a report on May 2018 of these dogs, being not in a leash, and they actually bit, this child. So, even though there's all those rules, but if people are not following them all the time, is there the potential for transmission, right? So the objectives of this small, project that I had for my non-thesis graduate student was to actually determine the prevalence of stools in dog parks in the metropolitan area of Birmingham, and we would use the traditional parasitology methods, right? So I teach them how to do the traditional parasitology methods. So why not just spend some time looking at dog poop, right, under the microscope? So we actually had 30 samples collected by one group of students in 2016. Then in 2018, we collected more, 40 samples. So 2016 was late summer, fall, early fall was the specimens of 2018. But my hypothesis, or our hypothesis, was that they will be all negative, right? And if you have done any traditional metaparasitological test, you know that is very tedious, especially in the US when you really don't have high prevalence of those parasites, right? So you stay there because you have to read the whole slide, and you might take 30 minutes just looking at nothing. Actually, a lot of things that don't look parasites, but it's not parasites. At the end, you wanted something to be a parasite, just so that you're just dreaming about parasites at some point. But so I have to give kudos to the students who's actually really wanted to do that, because they knew that they had, even though only 30 samples, only 40 samples, but they had their work cut up for them. And usually, you want not just one person to test or two, right? So I was thinking, the background parasitemia is already low. There's not many endemic parasites, there are some that are important, but it's not compared to other places. People like their dogs, they're taking them to the park, so they probably are taking care of them too with the other preventive measures. So it will be all negative. So what we did is that, so my students went and just use Google and search for parks, dog parks. And in 2016, we selected 12 parks, and they are here, you know, in the area, and 30 specimens were collected. In 2018, there were 2 parks that were closed, or one was like in somebody's residential area in a condominium, so we couldn't have access anymore. So out of those 12, we just went and visited 10. So we collected 40 specimens there. So the lab testing, so we had our, you know, little vials with formalin to preserve the stools from collection to testing. We had gloves, the list of the parks and how many samples we needed from there, our little biohazard bags. And we use this method that we call the formal ether sedimentation concentration method. So if you think, you know, from the intestines are big, all that stool, you're going to take a little bit, and then you're going to put under the microscope 1 drop of that specimen. So you can see already that's why you have to really take your time looking, because it's not very sensitive, right? You're just getting a little bit. So one way to improve your sensitivity is just to concentrate a little bit more. So in that drop, you have a larger representation of the animal or the person's stool. And you use a centrifugation method. And at the end, you have a little sediment here, a little pellet that you can take a little bit of that, put under the microscope, cover with a cover slip, and then you can look for parasites. So what are the results? Actually, I was wrong. There were actually 3 positives on the first set of 30. So we had 1 larva, and we had 1 hookworm egg, and we have another larva that we found. And on the second set, we found 1 hookworm egg only, right? So from 3 out of 30, right? So it was around 10%. But then the other one, it was 1 out of 40. We actually combine all of these results. So here are some of the pictures from our sample. So we have this larva here. We also have, you know, the 2 eggs that really look like hookworm eggs, right? Like I told you, this is like simple, right? It's a simple study. It's like having a hypothesis, using a method and collecting data, trying to understand what can you make of those results, right? So it was not a comprehensive study, right? So it's a pilot study. We chose some parks, right? We didn't measure the size of the eggs and the larvae. That's very important in parasitology, because sometimes eggs will have the same, very similar morphology, but from the size, you can actually, it can aid you in the identification. A few samples from 2016, they were collecting from the soil, so I was worried about those larvae too. We were not able to culture them or anything to be able to get a final diagnosis. And we use this traditional testing method, right, with the low sensitivity. We also focus more on helminths and not on protozoas. You know, our students are starting, and, you know, we wanted to make it a little bit more simple for ourselves. So if you're not a parasitologist, you're thinking, why are you calling it hookworm, right? So if we look at the larva and at the egg, you cannot differentiate these species that cause, you know, they're a hookworm in humans. There is Ancylostoma duodenale and the Necator americanus. So you can see here the teeth for Ancylostoma, the plates for Necator. And so if I only have the egg or the larva, that's usually what I have in the stools that either the dogs or the humans pass, I have to call it hookworm, because I don't know if it is Ancylostoma or Necator, or in the case of animals, if it's other species, right? I would need the worm to do that, or other tests. And once we are looking at these eggs, sometimes, you know, we know that there are some other parasites that have similar eggs. Like, for example, here is hookworm, here Stricostrongyl. You just see that is more pointed and a little bit larger. And then we have strongyloids here, Fulleborni, that is actually a strongyloids of primates, because usually in humans, you see more larvae, very, very rare to see the egg, because the egg explodes inside the intestine already, and people actually pass the larva. So in here, we could find that, 5.71% of the specimens were tested positive for either the parasite or the larva. And actually here, we're thinking hookworm, right? So, it was for my surprise that some were positive. So, it was exciting, right? You want something to be positive. So we thought, okay, so what we can do next? So the next thing that we thought about is to actually do this again and see if we can find the same thing or more or less, but now add, also a molecular technique to detect, to diagnose this parasite. So in the second part, these others, we had 30 and 40 samples, right? But we lost the 2016 samples. That happens sometimes. Our freezer, just thaw, and we lost that. But we had, there's 40 samples that were collected in 2018. And then the students went and tested now using a molecular method, right? So our hypothesis at that point was that we will have more positives than by molecular testing, right? So we only found one positive from these 40 samples by the traditional parasitological method, the concentration method. So if we do a molecular test, if we test it by PCR, we were thinking maybe we can get more, right? Because the other one is very insensitive, and then the PCR is more sensitive. And a lot of people complain too about cross-reactivity. With molecular testing for parasites, it's not that easy also to develop, primers and everything, but there are some tests that are used, in clinic and everything. So we just went and looked, what are the species, right, on dogs that can actually cause a little bit of trouble for humans that can be considered zoonoses. And we look at papers that had primers that use that for PCR. Another thing, because we sometimes will collect, the first time we'll collect from the soil, but we got better the second time. So we were getting them from the trash can, from the bags, but just to make sure that that was a dog sample, we actually did one of our PCRs is actually to look at this mitochondrial NADH dehydrogenase, just to confirm that it's, from the specific species, right? We're looking for dogs. And then we had 3 primers that combined in sets of 2. They would either detect Ancylostoma brasiliensis or Canino and Ceylanico. So these 2 would have to have another step to separate between them, but at least we could test for these species. So in terms of our results, so this is our PCR for the mitochondria. So all of them were positive. So all of them were dog. All of them were dog. For Brasiliense, you see here. So this is just one. The control from this here, we just use as a positive control, but it's a mitochondria. These are all primer dimers, so these are all negatives. This size is supposed to be a little bit higher here. And then our PCR for Ancylostoma canina and Ceylanico, it was positive just for one sample. So what we did, we took that sample, the protocol in these papers was to do a digestion, a restriction digestion, but, for us, it was so much simpler just to take that gel and take that band and send it for sequencing. That's what we decided to do anyway, because we thought we were going to have, a small amount of samples. So then we did, and the match came for a canine. Right. And if we look at the different hookworms of humans and dogs, so for humans, the most important ones are Ancylostoma duodenale and Necator americanus, but then you have these ones, right, that are especially in dogs and other species too. So they usually cause what we call cutaneous larva migrans, where because it's not the main host in humans, right? So what happens is that the larva, it stays on the skin, right? So usually feet, because you're walking barefooted. So it stays there, and you can even see the tracks. After some time, it dies and everything. The problem is that it itches. So as people scratch, then they usually have secondary bacterial infection, right, from that, because you scratch, then you're breaking the protective layer of the skin, and then bacteria can get there and you can get infection. So usually they cause cutaneous larva migrans, but these canine and Ceylanico, they can also cause, the canine can cause an eosinophilic enteritis, and that usually does not have, much symptoms, but Ceylanic is the one that actually can cause patent intestinal infections. And if you can see here, the geographic distribution is different too for these species, right? Although canina is the one that is most widespread, and that's the one that was positive by our PCR, right? And just to, if my thing again, do you think this is something from the past? I just found this paper, right, that is talking about an anterior cancellostoma canina hookworm infection in South Korea. And you can see here how small the worm is, right? And here we have the teeth, and they are small, but they, they stay in the gut. And just with the human species, the problem is really anemia, right? Because it keeps sucking all the blood, the nutrients, the proteins from the host, right? And if you have a lot of these worms there, then now you are competing, the worm is competing with you for nutrients. So usually people have this big anemia, right? And especially children, that can be very detrimental to their development, right? So in terms of conclusions, we found 4 specimens, right, that were positive for parasite, either egg or larva, and 1 specimen out of 40 that was positive for PCR as Ancylostoma canine, right? So even this was a small scale, but we can see, or we can extrapolate that, more people going to the parks, more dogs going to the park, if the control measures that are set there are not met, are not followed, then there is a potential for zoonotic transmission. So what we need to do next, and we have done that with a group of students in 2020, but because of COVID, we couldn't do it. It would be to test those samples, now that we have this DNA, for other zoonoses, like for example, Giardia, right? So Giardia is classified in different assemblages, and they have main hosts depending on the assemblage there. So that's what we were thinking about doing, right? And unfortunately, with COVID, we were not able to do. So again, do you think there's hookworm in the US, right? In the past, for example? And yes, so this is a picture, right, of like a dispensary or a mass vaccination clinic for hookworm, a mass treatment, sorry, not vaccination, even though there is people working on vaccines for hookworm. But this is 1910, so it's in the South USA. This is Arkansas, I think. Yeah, so Arkansas, and you have, people coming. So it says here, county dispensary for free treatment for hookworm disease, right? So the hookworm has a long history. See, 1550 BC, describing the, Egyptian papyrus. So all the way to now, where there has been a lot of investments, right, to make sure that it's eradicated. In the South, it was eradicated by, the donation of the Rockefeller Foundation, and all it took was really to make latrines or, kind of very rudimentary restrooms so that people can, the sewage would not be spread everywhere, right? And then people, if one person is infected, then, if then everybody else around can get infected. And then, there is this thing, there is this very nice video that says how a worm gave the South a bad name, right? So, because of the temperature and all of this, there were conditions in the South that really made it very easy for hookworm to disseminate. These parasites, they do not really discriminate, right? So if you have the host, if you have the parasite there and you have an environment that is conducive to the transmission, then you have it, right? They don't know borders, they don't know anything like that. They just want to survive, right? So some of these statistics is in 1905, 40 or more of the southern population was infected with hookworm, right? So this is a lot of people, right? So, and it took a long time to eradicate that with better sanitation infrastructure. It's changing those things, but it's more infrastructure, right? These people were getting sick, not because they want or not because of anything. It's the conditions that they had, in the environment that were they were allowing for this transmission to happen, right? And remember that I told you that the hookworm, suck all the suck proteins, suck all your nutrients from your blood. So if people are anemic, right, you usually are slower, right? You don't have so much stamina. You cannot really do too many things and everything. So then people took that and thought that people were lazy, right? What is not correct and is not nice at all. But I am originally from Brazil, even though I'm American now. But even in my doctoral, in my first master's thesis, because I work with another parasite called Schistosoma, I put this quote from this book that is from a very traditional Brazilian writer, and he has this character that he calls him Jeca Tatu. And he is someone actually with hookworm, and in English, I'm going to try my best to explain to you this very neat literary thing that the author did there, because in English is only the verb to be. But if you learn Spanish, you know that, está and ser, right? So it's two forms. It's either being like in a permanent stage or being in a transitory stage. So that's what he says here, that Jeca Tatu, he's not like that. Like it's not a permanent characteristic of him, but ele está assim. So he is like in a temporary, because of the worm. And he says here, right, that all these bugs, animals inside, is that what makes him ugly and inert and very lazy, right? And then I found this cartoon here, the story of a boy, where you have here, right, and the dog is here, the foot, right? So you usually have the infection, and it says, oh, you hookworms, right? So it was very interesting to see that in two different cultures, people really associate these things with laziness and everything, and they have a physiological reason for feeling that way because of the anemia. And it has to do with the cycle of the parasite and the way the parasite is trying to survive. But all of this, it's really because of the environment that these people actually live. So do we think that there's hookworm in the US in the present, right? So that's another question, right? These are old books and old things, and I showed you old pictures, but what about now in the recent present? So here it's some data from 2018. So here's our map of our beautiful state of Alabama, and Lowndes County is right here in red. So people, we started to do surveys, and I'll show you the publication, looking if there was hookworm, and there are some areas in Alabama where people, the septic tank, it costs as much as the annual income of the person. So if people get $15,000 per year, but the septic tank costs 12,000, then there's no way you can afford that. And also they would live in areas where you have flooding, right? So you have the sewage in front right here, and then if you have rain or if you have flooding, then all the sewage will just go and be everywhere, right, where people actually can step. They have to pass by those to to come out of their house and get into their house. So here's Butler County. It's just below Lowndes County, and here's the publication, right? So human intestinal parasites burden and poor sanitation in rural Alabama. So a lot of authors here, and if you are following COVID very closely, then you have seen Dr. Peter Hottis, right, from Baylor College of Medicine, talking about vaccination and talking about COVID vaccination specifically. So he's a tropical medicine physician and researcher. So what did they found there? So they surveyed 24 homes, right? They saw that 42% of the people there said that they were exposed to untreated sewage. They were able to get 55 fecal samples. Usually people do not like to give, to donate or to give fecal samples for research, for public health. I know that from experience is hard for people to do, but they were able to get 55. And here they found 19 that were positive for Necator americanus, right? Although, because they tested by DNA. So they didn't test like my students did in the beginning for the traditional method. That is considered the gold standard, unfortunately, for most of these parasites. So they found, but in very low levels of DNA, what we can, sometimes that's what we want to make they have an idea of how many worms of the worm burden, right, the parasitemia based on the DNA level. So it was low. So those people, right, if you believe this data, then they will be infected, but not highly infected. It will be a low level infection too. They also found 4 with strongyloids, 1 with entamoeba, that's protozoan. They also had some soil samples. They also did serology, and they found 1 positive for Toxocara. That is, askers of dogs and cats. So it's lacestod. So because this went into the news, right? There was this paper published. So of course the local health department has to follow up. And they did do an investigation. They were able to get 9 specimens from these, these 20 people here that were positive, and they did the ONP. That is what we call the ovine parasite examination, is the name of the test, the traditional test for diagnosing parasitology, and they found them all negative. So now, you're thinking, I hope you're thinking, do you believe that? What happened? So we know that test, it's really not sensitive, right, to the traditional parasitology. So even if the DNA is already saying that people have low worm burden, so to find those eggs of hookworm or larva, it might take a long thing. So so in terms of the public health, it seems like, so the conclusion is that we don't have an outbreak or something like that. But then we have this publication that actually shows some evidence of the parasites right there. So here's a picture. I'm sorry of the gross pictures and everything and talking about stool all the time. I mean, if you're a parasitologist, you're totally okay with that. If you are my student, you know I gross you out anyway. And there's very limit. My limit to that is very, you know, sometimes I'm like, I'm not going to put that picture, and they students go, oh, there's a limit. You know, I said, of course, everybody has a limit to their grossiness. So, you know, here is an interesting case, and it was from 2018, where this 17-year-old boy, he went to Florida, you know, on a church trip, and out of the 17 people in the group, 7 contracted hookworm, and you know, he's here in the soil, right? Maybe there's some dogs on the beach and they poop there, and then you have all of this going on, and here it's his foot. Unfortunately, this is what we call it the secondary, you know, infection, right? So you scratch, and then there's bacteria that get there, and then you kind of have to treat, you know, that now. So that is what I had to share with you guys. But this, I wanted to acknowledge, you know, Megan is one of my students, former students. She graduated now, she's a professional. And my son was in high school, and I made him go collect dog poop in dog parks. But you know, he needed a project for high school. So here he goes. He went and helped Megan with that. He, of course, he went under the microscope too, but you know, he didn't have the training, just so that he knew. And then we had Joshua, Sarah, and Sajal that collected those other 40 specimens. And then my students, Ching, Brittany, and Brian, they actually did, you know, the molecular testing. So they all present, you know, this work in our poster presentations for our graduating class. Actually, Joshua, Sarah, and Sajal, they won as the best non-drug seeking poster or, you know, graduate project for our students for that cohort. I also have presented this in as oral presentation or as poster in tropical medicine conference in Brazil for, you know, infectious disease, and also in a public health conference here in the US for practitioners of the whole time. And one of the questions I got there is like, did you put a press release? Did you let, you know, I mean, you know, it's not, I wouldn't be confident in putting that out. Of course, I want everybody to be careful. This is like not even based on this study, is based on the, you know, the knowledge that we already have, that if you have animals and people together, if the conditions are good, zoonotic transmission might occur, right? But because we didn't do a sample that we can really, you know, have a representative sample, then we did not want to, you know, I mean, we are doing this for research, not really as public health officers, right? But I put this here just to encourage you guys to, you know, that sometimes it's a simple idea. It's something you're passionate about. You know, you have people, they're like-minded and they want to do that. You have a little bit of money, and then you do. And, you know, just I encourage everybody to really, you know, share with other people. We're writing those publications. So we have some references, and just because we were talking about dogs, this is the Oliveira pack. So this is our pack. So our first adopted was Atlas here from the Humane Society. Then we got Apollo. And then my daughter found Astor on the street. Astor has been actually with another family now. And then we got Laura now. So, you know, they keep us busy. They're a lot of work, but, you know, they are also very sweet, and, you know, we love them so much. And if you have any questions, I would, you know, be happy to take them at this moment. And thank you so much. Thank you so much for watching.
Thank you, Anna, and thank you for the scramble at the beginning of the webinar that didn't go as planned. but we got through it, and in keeping with the not part of the plan, it looks like Lauren has dropped out. She was experiencing internet issues, so perhaps it bit her again. I will start with the questions. We've got a couple here. Melissa is asking, have you correlated the location of the parks with positive results with any type of socioeconomic barriers?
Yeah, that's a great question. And no, we didn't. You know, if we, you know, it's just that we went, we would have to see where we put those lines and everything, but it might be that for specific areas, we only have 1 or 2 or 3 specimens from there, and we have very little, but that is a great, you know, it's a great thing to kind of do, because, you know, that's the thing too, the thing with, you know, disparities and things like that. And sometimes we want to put people in a classification, but, you know, when we go to the bottom of it, it's, you know, most of the time it's not physiological. The differences is they are really socioeconomical. So I think that would be great. Yeah, but I haven't done that yet.
Well, I want to jump in with a question of my own, since it relates to this. First of all, is it feasible to perhaps use the trash cans at the various dog parks as sort of pool testing, and then be able to run PCR on the pooled samples to get an indication of whether this is an area that's, you know, like a COVID-19 hotspot, and end up sort of ranking parks based on that?
Yeah, that's another great idea. You know, the pooling for the traditional test will not be a good idea, because we already concentrate to kind of see a little bit more to improve that sensitivity. But for PCR would be a good thing. That will be a way to quickly improve our, our sample size. Yes, that's a great idea.
William Benjamin's curious if the PCR positive specimen, the one with the, or was the PCR positive specimen the one with the ova?
Yes, Dr. Benjamin, a true parasitologist that I think is Dr. Benjamin here from our UAB, I think. If it is, he's great. I think he didn't mind at all me talking about stool all the time and gross pictures. But yeah, Dr. Benjamin was exactly that one. But we thought maybe, we would detect others. But so maybe my students are doing a great job at the traditional diagnosis of parasites, at least of helminths, because their eyes, even with a less sensitive test, was a very good correlation with the results of a more specific technique like PCR. Any other questions?
I have a question.
Sure.
So, and forgive me with my physicist background, because it's not my area, but when we took our puppy to the vet, and when you take the puppy to the vet, all the first puppy visits, they do tests for parasitic infections, right?
Right.
And they always came up negative for our puppy, but the vet treats for parasitic infections anyway.
Right.
So what I'm wondering is the traditional methods that you showed before, you went to the more sensitive methods for testing. If that's the most prevalent method that's being used in veterinary offices across the United States, could the incidence of parasitic infections across the US be vastly underreported, and maybe even vets kind of suspect something like that?
Yeah, I think talking with a friend of mine that is a vet, that's what she was saying. Although, both for humans and for animals, we use a concentration method that we call. But for humans, we use the Hoffman that actually, or that one that the Hoffman is just naturally sediment. But if you use the centrifugation, then you have with the chemical, then is the one that I showed you. So it's on the bottom that your parasites are going to be. The vets, they prefer another concentration method that is a flotation method. So actually the parasites will be on the top and all the junk gets actually on the bottom. It's a little bit more cleaner for people to see, right? So you have the top of the tube, you just put the cover slip there, you get whatever you have go under the microscope. But if the eggs are bigger, or if they have what we call like a little top that it's what we call the operculum. If they get water, then they get heavier, right? So with the flotation method, anything that is too heavy is not going to float. So that's one of the limitations. But I don't know why, and this is so curious, but why the vets really prefer the flotation methods, while in the regular diagnosis is actually the other one. But both of them would be concentration methods. But because the sensitivity is not that good, that's why my friend was saying that even if it's negative, we don't even report negative. We just say no parasites seen, because, we cannot attest that is really negative, right? We just didn't see any parasites. And even when you're doing parasitology, even for people, for humans, right, you want at least 10 days, 3 specimens within, every other day for a period of 10 days. You don't just want to diagnose with a negative first. If the patient still has symptoms, you want another sample, because, they have life cycles, and they might be at a stage of the cycle that is not, you're not detecting that specific life cycle in the feces, so you want to make sure also you increase your number. So that's why, for people, we would do at least 3, at least 2 if it's positive. But, and that's why for animals, you just do there, and the flotation is a little bit faster. But, if it's negative, because you have them, you still have the puppy right at the visit, and they have to make a decision before you leave, so so they would treat anyway. But it would be too much, because you treat those, you treat very easily, right, those intestinal parasites, for humans. And even though not every single parasitic infection is benign like we would think, there are some people that can complicate, especially if you're immune compromised, and a series of other things, depends on how much you've been in contact with. But most of the time it's easy to treat. So, because the medication is around, people just go ahead and treat, and, be with a clear conscience. And with puppies, we know that they have, sometimes even you can see them, you can see the worms, right, in the puppies' poop sometimes, we can see in the stools. And I see Dr. Benjamin here asking that I didn't find any Toxocara. No, we didn't. And Toxocara will be a very beautiful and a little colorful, it will be more another, but we didn't see any Toxocara. Not, it's hard to find a hookworm egg. Is not that simple, but they found it. So I'm really proud of my students as clinical diagnostic scientists, and also as researchers.
Anna, why isn't the PCR technique relied upon more, other than expense, of course?
Yeah, also expense, and also in terms of the, you have to have a specific equipment, right? So with this traditional technique, you just need a microscope, you just need a centrifuge, and this reagent. So any clinical lab, right, in a hospital, in a vet, is easier to have those than actually have a PCR machine and primers. And also because it's you require people who have the expertise to do the PCR, and how to interpret, and you would have to have the primers, and sometimes you don't have for all of the parasites. But for malaria, even you can do a PCR, and it's even counted as a case. That's why we don't know some of these. It's just, now I'm going to put my plug in public health, the diseases that are notifiable. are the ones that we know better about the numbers, because any positive needs to be reported, and somebody at a health department is counting those numbers, you know. But most of those parasitic diseases, some of the mycosis, they are not notifiable. That's the name we call them. So, you know, then there's no central place knowing all of those numbers, and, you know, people are not required to report those. So
That's really interesting. So maybe is there a case for an effort on the part of data science and people making all these data dashboards? Maybe
yeah,
like infections we need.
Yeah.
Yeah.
Yeah, another thing too, and now I remember, Maggie, is important also, is that it still has a lot of PCR inhibitors, so it's hard to standardize that too.
Okay.
You know, so you know that why most of the parasites are intestinal parasites, then you have to take that into consideration. You know, so it takes time to standardize that and be in a way that everybody can use. But you have some inhibitors there. So PCR is great, right? It's very sensitive when it works. So, but you have to make sure that you don't have contamination, but also all these inhibitors, right? And if you have hemolysis in the serum and things like that, so that's why it's in stool. Sometimes it's the genome of the parasite that has a lot of repeats or a lot of G's and C's. It's hard to design primers and things like that. So, but you know, in this case specifically is stool is, you know, it's hard. There are a lot of inhibitors there for PCR.
Okay.
Really interesting. Are there any other questions?
I don't see any more.
Okay. Well, I just want to thank you for such an incredibly interesting talk today.
Oh, thank you. Thank you.
We'll remind everyone. Go ahead. Sorry.
Yeah, the parasites rule, right? If there is a microbe there, you know, it makes things more exciting. That's why even it's because of a virus, right? That we are not even together in person this time, you know.
Yeah. Big difference in our day-to-day lives and lives of our little buddies too, our little pets.
Yeah, and it makes always life interesting. That's the thing. There's not a dull moment in microbiology.
Well, thank you so much. Before we go, I just want to remind everyone about our April seminar, April 21st, 2021 at noon. We have Dr. Zenithia Barnett, talking about crayfish and dams. So thank you everyone who attended today.